Welcome back to the pharmaphorum podcast. In this episode, I speak with Dr. James Field, founder and CEO of LabGenius, to discuss synthetic biology, robotic automation, and machine learning in life sciences. Artificial intelligence requires one simple question to cut through the noise, says James, and that is, show me the molecule. Looking past the hype and seeking the demonstrated approach of novel implementation of machine learning, this, he says, is the way forward in employment of advanced technologies in life sciences. Tune in for a conversation on raising the cognition barrier. Send in your comments if you have any. And, as ever, thank you for listening. Welcome to the pharmaphorum podcast. This is web editor Nicole Raleigh, and today I have with me Dr. James Field, founder and CEO of LabGenius, a drug discovery company pioneering the discovery of next-generation therapeutic antibodies. In this episode, we'll be discussing synthetic biology, robotic automation, and machine learning in life sciences. Welcome, James.
Thanks very much for having me on the podcast today.
So, therapeutic antibodies are a vital weapon in combating diseases such as cancer. But developing these treatments through conventional methods is slow and unreliable. This is because it relies on human scientists working to sequentially optimize each property, such as binding affinity, stability, and toxicity, with improvements in one property often de-optimizing another. So it was that LabGenius set out to solve this predicament. But before we sink our teeth into today's topic, James, perhaps you could share with listeners a little about yourself, your journey to today doing what you do at LabGenius.
So I entered the world of synthetic biology really at the end of my undergraduate. So I completed an undergraduate in biology and microbiology, and then had the opportunity to join a master's program at Imperial College London in synthetic biology. And that was really my first exposure to the discipline. And then after that, did a PhD in synthetic biology, where I really got the opportunity to explore novel methodologies for protein engineering. And it really felt like an opportune moment, because we were going from a world where experimentation was being done at low throughput, pipetting by hand, to suddenly a world where we had the option to use high throughput automation, to use the power of compute. The cost of gene sequencing and synthesis was really coming down very rapidly. And it felt like a really opportune moment to combine those disparate technologies to create a new way to engineer protein therapeutics. And really, it was at that confluence of different technologies that LabGenius was born.
Thank you, James. So let's now sort of plunge into the technology behind our main foci today. That is to say, of course, AI, or artificial intelligence. How can industry cut through the hype that surrounds AI and optimally assess the technical underpinnings of an AI proposition in medicine?
I think it's a real challenge, actually, because the field is moving so rapidly now. And every day, you just have to open up your web browser and you're hit with the latest innovations in artificial intelligence and machine learning. And I think it is a challenge for industry to try and pass all of that noise and really understand where's the signal, and more importantly, how are these different technologies going to impact, in this case, the world of drug discovery. I would say probably the easiest way to do that is to ask one very simple question, and that's show me the molecule. There are loads of different ways in which we can apply machine learning to engineering proteins. But at the end of the day, the only thing that really matters is, can you produce a better molecule that's going to have a higher probability of getting through clinical trials and having a positive impact on patients? And I would say, that's what it all really comes down to. Can you really demonstrate that your approach, your novel implementation of machine learning is going to create better molecules? Now, in the case of LabGenius, we really focused down on answering a really important question in the development of novel therapeutics, which is, how can we engineer novel therapeutic antibodies to distinguish between healthy cells and disease cells and very selectively kill disease cells? So in the case of the work we do, what we aim to show is taking validated industry benchmarks and showing how our molecules can perform well over and above those that others have created in the past.
Okay, show me the molecule. I like this very much. So before we sort of dig our teeth further into what you were just suggesting then, James, and we're going to be talking about next-gen antibody discovery more, and as we've been discussing, your field of expertise, synthetic biology, and also going more into the automation, I was wondering if you could now describe and explain the so-called cognition barrier and how LabGenius overcomes that by using mathematical models to understand how molecules will respond to disease and then design novel therapeutics with the right collection of properties.
Absolutely. And I think we can, start this train of thought just by thinking, what is it that's special about human cognition? Well, human cognition really is just a product of evolution that's here to allow us to be successful in the macroscopic world and successfully reproduce, pass on our genes to the next generation. What human cognition has not really evolved for is how you engineer molecules at the micro and nanoscale. And so actually, when we think about the cognition barrier, we're actually looking at a very low bar here. When I was doing my master's and PhD and postdoc engineering proteins, something that really struck me was pretty much everyone is a bad protein engineer, by which I mean that the link between a protein's design and its function very rapidly becomes quite non-intuitive as the complexity of the molecule increases. And that's really not surprising because we haven't evolved to engineer proteins. But at the time, the way in which proteins were really engineered were by experts looking at crystal structures on screens, rotating them around and trying to work out what changes that they should make. And so for LabGenius, the breaking the cognition barrier really comes down to how do we build a platform that's able to systematically generate the right kind of data that's pertinent to the biological questions that we want to ask, build models from the data to really explore very large areas of design space, and ultimately to find really high-performing molecules that you would never have found using conventional methods. And the beauty of this approach is you can start to answer some of the most interesting biological questions. And one thing I would emphasize here with the approach that certainly we take at LabGenius is a lot of the time when you're using machine learning, the data doesn't exist in the public domain, and you actually have to generate it in-house. And that's why if you want to build a smart robotic platform that's capable of engineering these molecules, it's not just about AI, it's about bringing together a platform that can generate the data, systematically store it, analyze it, and ultimately explore design space.
The whole 360, as it were. What about co-optimization now and the possibilities of bringing more therapies to market as regards everything we've been discussing today?
Yeah, so there are two things that you kind of really need to take into account when you're engineering a novel protein. And the first is, does it have the right function? And often that in itself can be a really, really hard engineering problem, especially as the complexity of these antibody therapeutic molecules increases. And the second question you have to ask yourself is this molecule going to be a great drug? And by which I mean, is it highly developable? Are you likely to be able to manufacture it? Is it going to be stable? Is it going to be immunogenic? And historically, these questions have been answered sequentially. So you optimize for function first, and then iteratively try and optimize for the other features. And as you rightly said at the beginning of this discussion, the challenge there is if you're running that sequential optimization process, optimizing for one thing can then subsequently de-optimize for another. So the beauty of taking this ML-led approach is what it allows you to do is you're able to generate many, many different drug designs, physically build them and test them in the lab, and then using the data that's not just data relating to function, but all those other properties that you care about as well, you can independently train models that are predictive of those different features. And once you have those models in hand, you can then optimize across them. And what that really enables you to do is run this simultaneous co-optimization process where you're able to end up with high-performing molecules that don't just do what you want them to do from a function perspective, but also have all of the other key drug-like properties that make these molecules likely to do well and progress towards the clinic.
Okay, so if we think of examples now, if you're able to give examples. I mentioned at the start of this conversation that these therapeutic antibodies are vital in the oncological space. I was wondering if you wanted to share with listeners any examples where LabGenius's technology has been progressing in this field at all.
Absolutely. So, one of the key challenges that we've tried to tackle with our platform is this issue of recognition. So with conventional antibody design, it's often the case that these molecules are simply designed to bind to a given target of interest, and then in the case of oncology, often to kill the cell that bears the antigen. Now, that's a real challenge if you're trying to take on solid tumors. And the reason for that is a lot of the tumor-associated antigens are present both on the disease cell and also on healthy cells as well, albeit at different levels. And the big challenge there is you have a lot of healthy cells being caught in the crossfire. Now, if you're a patient, what that means for you is often, you might suffer horrendous side effects because these antibodies just can't distinguish between your cancer cells and your healthy cells. So what we try to do at LabGenius is to re-engineer these molecules by making them more complex, to be able to take in it's a form of molecular computation, if you like, where the molecule itself can take in multiple input signals and then make a decision over which is a healthy cell and which is a disease cell. Now, the scale of the engineering complexity here is an order of magnitude greater than for a conventional antibody engineering project, which means the link between these molecules' design and their function are extremely challenging and not really tractable from a human-led design perspective. But this is a problem space that you can start to address using this ML-driven approach. So just to give you some concrete examples there, we've shown across multiple tumor-associated antigens by taking this approach where the molecule itself is able to take multiple inputs. It's called an avidity-driven selectivity strategy. We're able to engineer these molecules so they have complete on-off killing selectivity for tumor-associated antigens where the delta, the difference between expression levels between healthy and disease cells, is actually very low. And as far as we're aware, we're the first group to have been able to show this. So we have some extremely promising molecules now that, again, can show the ability to completely distinguish between these healthy and disease cells. So they're entirely selected just for the cancer cells. And by engineering these molecules so that they can then recruit immune cells to kill your target cancer cells, these are immune cell engagers, we have real confidence that this offers a paradigm shift in the way in which we might be able to tackle solid tumors.
Thank you, James. So my last question, and they're pretty much the same throughout this series, what of the future horizon from the 5-year view to the 15 for LabGenius?
So I would say that the trend here that we're seeing is how do we engineer molecules to be more complex? And I'll go back to this kind of idea of molecular computation. First generation antibodies, really the way these molecules worked is they just found a target antigen, they bind to it, and they exert their therapeutic effect by that mechanism of action. These next generation therapeutic antibodies really are quite complex in their ability to take in multiple different inputs and then effectively make a decision over which pathways to antagonize or agonize. And I see our ability to kind of play with that molecular complexity only increasing as the experimental throughput of these high throughput platforms and these new machine learning methods get combined and integrated and ultimately improve our ability to create and grapple with molecular complexity. So I think over the next 5 years, we'll see a whole raft of molecules that have improved properties like the ability to be more selective, but also to be highly developable as well. And then over the sort of the slightly longer term time horizon, we'll see the deployment of molecules like this across many different therapeutic areas. And indeed, we're starting to see that now as well, actually, with molecules like T-cell engagers starting to entering into the field of immunology, where the requirements are similar, but the bar is somewhat higher in the sense that if you're going to dose these molecules chronically, then of course you have to have a slightly different safety profile. So we'll see a big expansion of the applicability of some of these complex antibodies across different therapeutic areas. So I would say those are probably the two big themes that we can expect to see over the next few years.
Thank you, James. It's been a pleasure.
Thanks very much for your time. I've really enjoyed the conversation.
And so that concludes another episode of the pharmaphorum podcast. You can find more information about this episode, including a download link and information about previous installments at pharmaphorum.com/podcasts. The pharmaphorum podcast is also available on iTunes, Spotify, Amazon Music, Podbean, and pretty much wherever you get your other podcasts. Just search for pharmaphorum and press subscribe. Of course, don't forget to visit our website itself, where you can sign up for daily and weekly news and analysis bulletins. And do follow us as well on X or Twitter and LinkedIn at pharmaphorum. That's all for now. Thank you for listening.